Shirley, are you there? Shirley,
Yes, I'm here.
please remind me, or somebody please remind me about do the review
Sure, just give me a second.
for the quiz number 1.
Okay.
Okay, so I want to review the answers, etc. Okay, so let's start with we talk about cells and tissue and membranes. Okay, so that is again part of bioscience review, and we are going to go again topic by topic. Okay, so we will talk about. I show you. We are talking about cells first. What are the functions? And even though those are basic biology, I'm going to do some approach on nursing considerations, and I will just elaborate one by one. All right, so we are talking about microscopes, and yeah, we are going to talk about the lens and the plates and all that. No, 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 not at all. So I just want you to realize about the study of some microorganisms. That is called the cytology. Cytology is the study of cells. There you are, a study of cells. Okay, and you know, a cell, one cell, is going to be the basic unit of life. So remember, we was talking about the functional unit. So the cell is called basic. Is basically the functional unit because a cell, you as a person, you eat, you drink, you pee, you poo, you breathe, right? You reproduce, you grow, and we die. Correct? So that is exactly the same that is doing a cell. A cell is need to drink, they need to eat, they need to pee, they need to poo. Right? So they need to reproduce, they grow and they die. So this is the functional unit, or we call the basic unit of life. They can reproduce, right? The cell can reproduce. Remember the cells we was talking about in bioscience, the cells, your cells of the skin, for example, if you have an injury in your skin, the skin is going to regenerate. This regeneration means that the cells are going to divide, reproduce in order to replace the cells who are being dead, was dead, right? So basically, so that is telling you that these cells are going to be the basic unit of life. That is in humans, but what about bacteria? The bacteria are unicellular unicellular living organisms. And you already know that living organisms are considered those who can multiply by itself. And bacteria is going to be under this category too. So the bacteria is a living organism. The bacteria is going to be like you and I. They need to live and they eat, they drink, they pee, they poo, they make gas, they reproduce, they grow, they die. So all these functions are going to be in this unicellular or each cell, including the human cells. All right, so the microscope are going to be used. Micro means small, tiny, and do you ask yourself how small things you can see? Just to give an idea, if we have here, for example, 1 meter, this is 1 meter. 1 meter are going to be divided in 100 centimeters, correct? Yes or no?
Yes.
100
Yes.
centimeters. So 100 centimeters are going, each centimeter is going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 millimeters. So each of these, one piece of this is 1 millimeter. Now you divide this millimeter by 10, 10 times 1 millimeter, 1 millimeter divided by 10, that is the maximum ability for you to differentiate a structure. Smaller than 1/10 of 1 millimeter, you cannot see it. It's not, it's your eyes, our eyes are not able to do that. So for that, we need to use the microscope. Okay, so and these, all these organisms that are under this size are going to be called microbes, right? Microbes. So that's why we are going to use with microscope. Okay, so these microorganisms, we have one cell in the body that is the largest cell in the body, the largest cell. This largest cell is the ovum and the ovary. So the ovum is the largest, the biggest cell in our body. This cell is going to measure about 0.1 millimeters. So that means it's exactly the same distance or same size that we can see with your bare eye. Yes. In conclusion, you can see the ovum with your bare eye. Yes, one ovum is the only cell that you can see with your bare eye. It's very big. It's about 1/10 of 1 millimeter. So that is the maximum capacity of human eye to visualize and structure. The spermatozoid is going to measure 20 times smaller. 20 times smaller. It's like you're walking in the street, you find a building of 20 stories, 20-story building. You are walking on the lobby, you are the spermatozoid, and the building of 20-story is the ovum. So that is the difference. Okay, so let's keep going. Okay, cell. Cell is an autonomous, self-replicating, independent and functional unit of life. Functional unit of life is the minimum component that hold the whole characteristics of you. So everything that you're doing, the cell itself, one cell can do that too. Okay? So we are composed by multicellular, so we have about 100 trillion cells in our body. 100 trillion. 100 trillion is like 100 million times a billion. So it's a lot of number, right? So, and remember, please, each cell of your body are going to need to do everything, right? Inclusive to consume energy. So every time that you're eating something, that is going to go to the 100 trillion cells in your body. Okay, so oxygen, elimination of carbon dioxide, all the nutrients that you eat are going to feed 100 trillion cells. So you have a population, you're responsible for 100 trillion people, 100 trillion cells. All right. Okay, so we will be discussing the anatomy and the typical cell in the body. So that is a review. So that's why you're going to get familiar with that. So I expect some questions. All right. So here we have this cell, and this cell you have all the characteristics of a cell. All the components. You already know that this what is inside the cell, including the nucleus, are going to be called organelles. Remember the division, the organization of the body: cells, molecules, cells, tissues, organs, systems, right? So the organelles. These organelles are going to be the mitochondria, the nucleus. All this is to an organelle, including the nucleus. Okay? So for this, you're going to use this table at home, and you're going to see by number the characteristics of each of them, just to review of them. But we are going to talk about one by one. All right, so when you see these lights here, most likely coming for the exam. This is for the midterm or the final when you see fire here. Okay. Largest organelle in the cell. The largest organelle in the cell will be actually the nucleus. The nucleus. There are going to be other structures are going to be the chromosomes, the nucleolus. So, but the nucleus is basically the largest organelle in your body. The nucleus. The nucleus is going to be composed inside by chromosomes and nucleolus. So those are big too. But the whole together, the nucleus is the largest organelle in the body. You okay with that?
Yes.
And the nucleus is going to contain the nucleolus and the chromosomes. Okay, so just to review here, when you have a cell here like this, we have here the nucleus, and we have the nucleus, and the DNA is like that, right? Like a spiral, double spiral. And here we have, in other cases, we have the chromosomes. 46 chromosomes, correct? So what is the difference between this DNA and these chromosomes? The DNA is going to be in a spiral shape like this, a spiral like this, when it's not reproducing. This DNA, if you put it, this DNA, I put it like a stretcher out, the DNA like this, I stretch out the DNA like this, and I cut pieces. Each piece is going to be a chromosome. Each piece is going to be a chromosome. Each piece is going to be a chromosome. So when is going to be A, when is going to be B? The A is going to turn into B when it's going to multiply the cell. Are going to multiply. And A is when it's resting, when it's not resting completely because they're doing all the functions that they need to do, but it's not multiplying. We okay with that?
Yes.
Yes, Doc.
Yeah.
Okay, perfect. Excellent. All right, so let's keep moving. So we have the plasmatic membrane. The plasmatic membrane, plasma membrane, or cytoplasmatic membrane, cytoplasmatic membrane, or called plasmatic membrane, or cell membrane. So they have different names for the same thing. Okay, so similar, but you can call either way. So the plasmatic membrane or cell membrane is going to be composed by a bilayer structure. Bilayer structure. Here we have the bilayer structure. So please, just remind you for bioscience, I want you, because we are going to use that later, is that the bilayer structure is the main components of the cell membrane. Main components are going to be what? Who can tell me?
Phospholipids
Keep going.
Protein.
and proteins.
And proteins. Excellent. Okay, so those are the main. I didn't say the only components. I said the main components, so don't get confused. It's the main components. And the main components of the cell membrane will be phospholipids and proteins. But those are not the only one. Okay, but the main is PP, phosphoproteins, the main component. But there is other components that are going to have important function too. For example, we have in the cell membrane, we have this, for example, the cholesterol. Cholesterol is fat. So cholesterol is going to be immersed in between this cell membrane. Can you see this like a X here? This like a pink, those are proteins. Then we have here some chains of carbohydrates. Carbohydrates are here. These carbohydrates. These carbohydrates with a protein basically are going to be part of what we call the receptor for allergens. Yes. So allergens, for example, just give you an example. Allergens is coming, pollen, and it's going to be attracted by these carbohydrates and linked to the protein. So that is going to trigger out the release of histamine, for example. Okay, that is an inflammatory substance. Well, anyhow, so just remember, the cell membrane is composed by a bilayer structure. Bilayer structure will be the phospholipid. This is the head of the phospholipid, and we have one fatty acid, and another fatty acid here. So this is two fatty acids, one plus one molecule of phosphorus is going to give you what we call the phospholipid. Is that clear?
Yes.
Yes.
Okay, so this cell membrane, as we talked in the previous course, we are going to have what we call the transportation. transportation will be, we have passive, and we have the passive, and the other one is the active, correct? In the passive, we have the simple diffusion, we have the facilitate diffusion, and we have osmosis. And everything that is the transportation, the active and the passive, are going to be through the cell membrane. Simple, we have the concentration, for example, high concentration of oxygen, they can pass through simple diffusion. They're going to say, excuse me, I'm going to pass, in between the phospholipids. So they pass from out to inside the cell. The simple diffusion. Then we have the facilitate diffusion. Facilitate diffusion is they pass through these channels, these proteins. Proteins are like the doors of your house. They knock the door and you open the door in order to come in, right? So the same thing happened with for example, the monomers of the carbohydrates, like monosaccharides, the amino acids, and the fatty acids. So they're going to knock the door, and these proteins are going to open the door, and they pass the amino acids, the monosaccharides, and the phosphor and the fatty acids. So this is through facilitate diffusion. That is review. We already talked about that in that time, right? So we have the best example of simple diffusion is the gas exchange. Remember, passing oxygen, carbon dioxide. The best example of facilitate diffusion is going to be the absorption, because they're going to pass the fatty acids, the amino acids, and the monosaccharides. Okay, facilitate diffusion. You okay with that? Yeah.
Yeah.
Now, the cholesterol is, why is cholesterol there? Okay, cholesterol is there because that is, I want you to think about the cell membrane that is not really like a rigid, like a board, like a table. It's not like that, strong, or rigid, like a hard. This is like a wavy. It's wavy. It's not really like a box, like a concrete or like a wall. It's very flexible. This flexibility is like a silicon. You see a silicon, the movement of the silicon is similar of the movement of the cell membrane. And for that, we need to have some structure that give rigidity, stiffness, a little bit more stiffness to the cell membrane. Otherwise, will be totally gelatin, can be destroyed. And who is doing this stability, stable consistency of the cell membrane is going to be the cholesterol. As I told you in previous course, I'm going to repeat now. If you put your hand under the table, can you put your hand under the table? Now, do you feel that gum under the table? Somebody, once in a while, when you were somewhere, you feel that gum under the table, right? That gum is exactly the same consistency of the cholesterol. So cholesterol is not like fat, loosey fat. Cholesterol is like more harder. It's very hard. And that is what is immersed in the cell membrane. You okay with that?
Yes.
Mm-hmm. Okay. So the cytoplasm. What is the cytoplasm? Cytoplasm is everything that is, listen to this, very careful, cytoplasm is everything that is in between the cell membrane of the cell and the membrane of the nucleus. This is the nucleus. Cytoplasm this is the nucleus here. Cytoplasm is going to be everything that they're going to mark here. That is cytoplasm. Everything is cytoplasm, but not the nucleus. Okay? So everything that is in between the nucleus and the cell membrane, that is cytoplasm. Okay? And the cytoplasm is going to contain mostly all the organelles. There's another term that is called the protoplasm and the cytosol that is included in the nucleus. So I'm not going to go on that. Never is going to be asked. But I just want you to be sure. What is cytoplasm? Cytoplasm is everything that is in between the cell membrane and the nucleus. Easy, right? You okay with that?
Mhm.
Okay, perfect. So here we have, this is a plant. A plant. This is a plant cell. Plant cell, think about your broccoli, think about your asparagus, think about your whatever fruit you like, or fruits, mango, I don't know. I just bought mango, banana, oranges, strawberries. So all those are plant cell plants. So what is important about this? Look at it. Here we have the cell. Okay, so they have the chloroplast. I'm not going to talk about is the photosynthesis. It's not a class for today. It's not about. Here we have the nucleus. Don't get confused. Look. This look like a nucleus, but it's not. It's the back wall. Back wall. For example, if you eat a strawberry or orange, a banana or whatever, when you crush the plant, this back wall is the one who contains the sugar. This back wall are the storage area. It's like in your house you have a storage. You have a storage. That is the back wall, and in that storage you keep a lot of sugar. That example, right? So you can be the vitamins, etc., are there. But when you crush a fruit, you break down the back wall, and the taste of the fruit is going to be here. Could be sugar, could be vitamins, etc. Okay, perfect. So now this cell of the plant contains a nucleus similar as our cells. In addition to that, I want you to pay attention to this. Where is the cell membrane? I will try to make it yellow here. Cell membrane here. This is. Can you see this small line?
Yeah.
Can you see this small line here?
Yeah.
Can you see this small line? Small line. This is the cell membrane. This is the cell. Oh my God! See, I have good pulse. I can still do surgery, I guess. But yeah. Okay. So that is the cell membrane. The cell membrane of the cell. But here, what is this? Can you see this structure? Very hard, very thick.
Cell wall.
Yeah.
Exactly. That is the cell wall. There you are, the cell wall. What is the composition of the cell wall? What is made of the cell wall? What is made of the cell wall is going to be composed by cellulose. 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 What is cellulose? Cellulose is a polysaccharide, or no?
Yes.
Okay. All right. So see, this lecture is the purpose of that. So I'm not pretending to remember everything 100% in the past. So that's why they repeat these lectures in order to refresh, and the information is coming to you again. So why is important to know about the cellulose on the cell? Because cellulose, remember, never forget that, is a polysaccharide, similar as the starch, similar as the glycogen. Glycogen is a polysaccharide too, right? Cellulose, amylopectin, etc. Right? So cellulose. Cellulose, what is cellulose? Somebody can tell me, what is cellulose?
Is it fiber?
Polysaccharide.
Excellent. Polysaccharide, and it's a fiber. That is the dietary fiber. The fiber. That is the fiber. Fiber is going to be so important. You need to eat about 25 grams of fiber every day, like 1 cup of fruit. That is to complete your diet of fiber every day. You need to, fiber is going to be the formation of the stools. Is going to prevent you to have colon cancer, prevent constipation. Fiber is going to be participating in the flora. Is going to give the food for the bacteria, and the flora is to protect your intestine. And you remember with bioscience that the flora produce vitamin K, that is for coagulation. So that's why we need fiber. Just remember, cellulose is equal to, say, fiber. Cellulose, if you want to do more specific, cellulose is the dietary fiber. Dietary fiber. Dietary fiber. Fiber is part of our nutrition. Without fiber, we will have serious problems. Okay, fiber, cellulose, fiber. We okay with that?
Yes.
Okay. So fiber is important in our diet. Cellulose. Okay. All right. So let's start talking about. Let's see here. Some example here. Here we have the Golgi apparatus. Talking about the Golgi apparatus, you know, I'm going to probably make a draw here. Let's suppose that this is a cell. All this graph, all this frame is a cell. And here we have the nucleus. The nucleus I'm going to draw in purpose here is a double layer membrane. Huh? No, similar but not the same as the cytoplasmatic membrane. So that is the nucleus. And why I put it like this? Because the nucleus have openings. Try to use your imagination, 3D imagination. And you see here, for example, this one. Can you see here? Can you see the nucleus? The
Yes.
nucleus are going to have this thickness. It's a double layer membrane, and they have these openings. So what? See? Can you see this opening coming out? Coming out. Okay. All right. So that's what I'm drawing like that. So here we have the. We are going to talk about the endoplasmatic reticulum. The endoplasmatic reticulum. Endoplasmatic reticulum. The endoplasmatic reticulum. The endoplasmatic reticulum. The endoplasmatic reticulum. So these are the endoplasmatic reticulum. Endoplasmatic reticulum. Now here we are going to have one structures here. You see that? Those are what? What are these? This is the Golgi apparatus. The Golgi apparatus. In the Golgi apparatus, what happened, you remember when we have the DNA here, this is the DNA, we have the nucleolus, nucleolus that contain the RNA, this is the DNA. The RNA is going to open, and there is going to come the nucleolus, and the DNA is going to open. So instead to go like this, it's going to uncoil, and they're going to do like this. So they're going to open a piece of the DNA, if you remember that. So this is open. And the RNA is going to copy the DNA. So that is called that transcription. Then the transcription is going to be the RNA messenger who is coming through the endoplasmatic reticulum. And here is where it's going to be the translation, where all the amino acids are going to follow the sequence of the RNA messenger. That is the function of the endoplasmatic reticulum. And then they are going to form what we call the primary protein, the secondary protein, the tertiary protein are reaching the Golgi apparatus, and the quaternary protein is going to be totally a functional protein. These are Golgi apparatus function, in conclusion, is going to help to get mature the protein. So the protein here in the Golgi apparatus will be quaternary protein or tertiary protein. So these proteins, then they can go out from the cell, or they can stay according to the needs of the cell. You okay with that?
Yes.
Yeah.
Yeah, it's coming to your mind everything about bioscience, right?
Yeah. Yes.
Okay, perfect. So that is the Golgi apparatus. So here we have the Golgi apparatus. What we have here, we have quaternary and tertiary protein. So, and this is going to be functional protein. You remember a protein becomes functional when you have a shape. So just imagine, just to review again, the quaternary protein is full functional. Tertiary protein, most of them are functional, not everybody. Secondary protein are not functional yet, do not have a shape. The primary protein do not have shape, do not have function yet. So the function is going to be like the lock of your door in your house. The lock of your door in your house. So you put the key, and this they have a shape inside the lock, right? So this shape is the protein. The key is going to be able to open the protein if the door is actually working, the lock. So they need to match. So that's why we need shape of the proteins. So this key will be, for example, some hormones that are going to recognize to open. So not everybody is going to open your door, right? The only one who has the key is you, all right? The key is the one who is going to open the door, the trigger. You are the hormone, the prolactin, the progesterone, the estrogen, whatever hormone you are. So you come into your cell, that is your house, you try to open with the key, and the keyhole is the protein who has a shape, a specific shape. So otherwise, everybody will be able to open your door. You're the only one, and you want that. You okay with that?
Yes.
Yes.
Okay. So here we have our friend, the cell membrane, and the cell membrane is going to contain these proteins. Remember, the protein, we have proteins in facilitate diffusion. The cell membrane is going to be two proteins, one like this and another like this, like a sandwich. Two proteins, one protein and second protein, two proteins. And the proteins, when they try to come here, the monosaccharides or the phospholipid or the amino acid, those are the monomers of the nutrient. They are going to come here and they are going to open like this. Open. This is outside, this is inside. They're open. They're open like this. This is the This is the monosaccharide. Oh my God. Okay, so this is the monosaccharide, this pen. This is the protein, my hand, two proteins that are going to do this. Open, like that. And then they're going to close here and open here. And actually, that is facilitate diffusion. What is going to enter like this is going to enter the monosaccharides. So please, the glucose, the fructose, the galactose are going to enter the monomers of the fatty acids. Fatty acids are going to enter like this and facilitate diffusion. And the amino acids too. The amino acids are going to enter with facilitate diffusion. So what is the best example? So if I ask you how the intestines are going to absorb the nutrients, they are going to be absorbed by facilitate diffusion. You okay with that?
Yes, Doc.
Yes.
And the simple diffusion is the gas exchange. So the simple diffusion is when the oxygen is said, the oxygen is so tiny that they're going to pass. This is like people here. I say, okay, excuse me, squeeze, I need to pass. And they pass in between them without any other protein, anything. Just pass from higher concentration to lower concentration. So the oxygen is needed in the cell. This is inside. We have low concentration of oxygen. You need oxygen, so the oxygen is high coming from the blood. And the oxygen is going to pass through simple diffusion to the cell. You okay with that?
Yes.
Yeah.
All right, so here, thank you. Here is the cytoplasm that you already know that is in between This is the space in between. It's in between the nucleus and the cell membrane. Here we have the endoplasmatic reticulum. This is the nucleus. Can you see the openings here? The opening, so that this is the double layer membrane of the nucleus. Here we have the DNA. They're going to pass the RNA with the transcription, and then the protein is going to travel all the way here, all the way here. They go to, and the endoplasmatic reticulum is having two components: the smooth, and this is the rough. Why is smooth and rough? We have here that you remember that we was talking about that here is where it's going to happen. It's going to happen that there's going to happen that translation. Remember the translation? The translation will be like this. So this is the RNA messenger with the codes after being copied by the DNA. So then they are going to bring amino acids. Amino acids are going to, according to the code of the RNA messenger, are going to bring the amino acids to form the protein. Amino acid, amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. It's a linear, a line amino acid. So a group of amino acids produce a protein. Now, who are bringing these amino acids to the RNA messenger? The ones who are bringing that, those are these dots. Can you see these dots? Those are the ribosomes. Ribosomes. The ribosomes. So if like you are in your house, you are in the nucleus, you are in the nucleus, your house is the nucleus of the cell. The city is the cell. The nucleus is your house. And the city, where you live, San Jose, where you live, Daly City, where you live. The city is the cell. Okay? And actually, when you want to make a recipe, you're going to go to in your house, you have the recipe. You copy in the book. That is the RNA messenger who is going to copy the recipe. Then you go out from the nucleus, and you go where? To Safeway, or to Laundrox, or whatever you like, Leonardo's or Whole Foods, whatever. So you go there, and you have the list of the ingredients in your hand. Then you stay there for a special service. Let's put it that way. There's people with carts. The carts, I say, oh, this is your list, and the carts are going to bring the ingredients to your cart. You don't have to move. You stay there. So they are going to bring a special service. They are going to put the ingredients into your basket, into your cart. So these guys who is bringing the ingredients into your cart are going to be the ribosomes. These ribosomes are the ones who bring the amino acids into the RNA messenger to produce that translation. And then you go home and you cook and have a nice dinner. Okay? We got it?
Yes.
Mm-hmm.
So
Yes.
these ribosomes are going to be located here in this area because they are going to bring amino acids to make that translation to perform the primary protein. But then after that, they are going to continue to this labyrinthic area. But this is already lack of ribosome. Why? Because we don't need it anymore. We don't need them anymore because you already complete the list of your ingredients. So they are going to keep traveling towards the Golgi apparatus. You okay with that? Here we have the primary protein, most likely here the secondary protein, some tertiary, and when they go to the Golgi apparatus, the protein becomes tertiary and quaternary protein. We okay?
Dr. G, can we
Yes.
take a break, please?
Who is this? Who is this? Shirley?
Yes.
Shirley? Yeah, sure, Shirley. Yeah, I was thinking about it. Yes, thank you for reminding me. So it's 11:05. We are going to have 11:15. Okay?
Okay. Thank you, Dr. G.
Thank you.
Thank you.
Thank you.
Just a moment. Where is this? Oh, O'Neill. What's your name, O'Neill? O'Neill again?
Kenneth?
Kenneth, please, could you please call me?
Yeah, I'll call you right now.
Sure.
Are you putting in our assignments for the homework as well? Right now?
What do you mean? No, I'm going to do it in the lunch break. I need 10 minutes.
Oh, okay.
Sure. Who said that?
That was Spa. Thanks.
Oh, okay. I'll see you then.
Is the homework due right now or is it due tonight?
No. It's before the quiz. So if you didn't upload it now because I should put it before the quiz.
Yeah, I have to do that.
Okay, so let's do it now. Upload it because if you didn't do the quiz, the purpose of the homework is lost. The purpose of the homework is to review for the exam.
Right.
Okay. I will come back at 11:15. Hello, everyone. Hello.
Hello.
Hi.
Hello.
All right. Okay. Hello, hello. Okay, guys. So I want just to cross some words with you for the whole group. I know that there is a time of adjustment for the first week, second week. We have the Proctorio that is new for you. The new directions for homeworks that are probably not clear, or actually we need to clarify more with you, is this. The homeworks that you must do is, for example, today is lecture number 2, correct? Okay. The homework should be delivered and uploaded in Moodle before the quiz on Wednesday. Okay? Why is that? It's not anymore today until 11:59. No. The homework should be given before the next quiz. So, for example, homework number 2 today should be delivered before Wednesday, the quiz of Wednesday. Why is that? The reason for that is that the summary of the homework, the purpose of this is you to be able to review again the lecture. If you're giving me the homework after class, the purpose of the homework is 0. It's not valid anymore. The homework for that is to review, to be able to review, to make you a summary of what we was talking in class, the previous class. Is that clear? We okay with that?
Yes.
Yes.
Okay. So that's why the homework should be given before the quiz. Now, for this week, I'm going to be lenient for that. So if you didn't do the homework, so please don't do the homework during my lecture. Don't do the homework during the lecture. I know that some of you are doing that now. So just stop doing that, because I'm going to give you extra time until today, until tonight, or only for this time. You okay with that?
Yeah.
Okay.
Dr.
All
G,
right.
I have a question.
Who is this? Miss Erica. Go
Erica.
ahead.
Is it gonna be the same for all homeworks? Is the summarization of all the lecture?
Yeah.
I mean, every
Yeah,
chapter.
yeah,
Okay.
yeah.
Thank you.
Miss Erica, I don't want you to give me like 10 pages summary. No. Half page or 1 page is more than enough. But what I want is each of you be sure that what you are writing as a summary is going to help you. That is the reason for the homework. Help you to memorize, to go faster into the concepts that we are going to talk in class. Okay? I'm sure that
Yes,
in
thank
the next lectures, that is going to be like an habit, and bit by bit. I know that the first week is kind of hard, and in anatomy physiology, everything is set in organized manner. And one more thing, the tutoring. The tutoring is going to start today after class. So 1 o'clock, we start doing the tutoring. Probably we can do it earlier than that, but that tutoring is going to be guiding you for your exam. So you have the study guide at the end of the course, of the website. I mean, at the end of the page, you can have all the topics that you must remember. And if you follow the study guide and you follow the concepts that we remark in class, you will be more than okay. So please, if you are conscious that you study every day 3 hours at least for the course, you will be more than okay. So there is no way to fail anatomy physiology, because this course is just AMP. It's not like a very kind of elaborate NASA going to Mars topic. No. It's just anatomy physiology. So, and I want to encourage everyone. So everyone, if you have some concern, just let me know. You're open to tell me everything in the manners that we want to understand each other. Okay, so none of you can be afraid to contact me or to tell me since how you feel about the course or an issues that you have in class or around. Okay? We okay with that? All right.
Yes.
So you can talk about this class for other courses, for other groups, and you will see that that is very much a time of adjustment for we are going to have. Okay? All right. So any other question, please? Any comments? Somebody? Alejandra, you have any question?
Not at the moment, no.
Miss Jessica, you have any question? Your camera, please. Jessica?
Sorry, no
Okay,
question.
okay. Shirley, any question?
No, Dr. J. I'm just a little like overwhelmed because I think that we didn't get a lot of good direction at first.
What do you mean? Elaborate.
I think it would have been a little nicer if we would have had like the clarification that we weren't able to go back. Like everything is just a lot different than when we had our bioscience class, you know, like when we would take the quizzes, we were able to go back. No one told us we couldn't do that. You know, so
Yeah.
it's just different. And I know it's not 100% honestly your fault, but I feel like overall we didn't get good direction when it came to like orientation. Like we kind of just got thrown into like evolve, and I feel like when we did our bioscience class, you gave us a lot of direction. You said, okay, this is how you do it. This is how you're going to do it. This is when you do your homework. You would do, you know, the study stuff with us after class. And it seems like everything is all over the place. Not you, but like everything in general is just kind of all over the place. So, you know, it's just a little.
Shirley, what is all over the place? Can you please let me know? Tell me. Go ahead. Be free, please. Don't be stressed.
I'm free. I will express how I feel. You know, like when we did our clinicals, it was like everything was all over the place. Like we went over things. Other instructors were being interrupted. We weren't being, you know, clearly explained things. We got an email from Dr. Sashi at 7 p.m. the night before, and the code that she gave us didn't even work to be able to watch the videos. And then we showed up to clinicals and we were basically talked to like if we were dumb because we didn't know how to do it. And how would we have known how to do it? Because some of us have never been in the medical field. I haven't, you know, and then to be expected to perform. It's one thing if we had access and then we didn't watch the videos, that's one thing, but we didn't have the accessibility to do so. You know, luckily we had people like Jessica and Thumb and, you know, people that had experience. So they were showing us how to do it. But we're expected to do things that some of us have never done before. I haven't been to school in 12 years.
Shirley, can you give me a second, please? Give me a second. I'm going to just do something. Just a moment.
Sure, no problem. Shouldn't have asked me for my opinion.
No, yes, he should. No lie, our conversation went well though.
Yeah, I did.
That's good.
Oh my gosh, I'm trying to do this evolve thing and it's asking me for something from the teacher.
Is it for fundamentals?
I actually
On the fundamental evolve is not working. I don't know. It's just not.
I
sent us a
actually
code.
messaged her. I actually messaged her for the code. I asked her for the instructor codes that it's asking us,
She
but she
sent it in
just
an email.
yeah, but that's not working.
Try both?
Yeah. Well, that's for the code was for the book to redeem the book, but
Yeah,
for the
but-
book itself, it's asking for the instructor code.
Yeah,
It does.
it's because we're having to figure everything out. Like,
Wait, what do we
I
need?
know.
I'm
It's
texting Miss
crazy.
Matthews. So what do we need? What isn't working?
It's saying talk to your instruction to find out if this is an evolve course or a course on Canvas provided by
Hello.
yes,
Hello.
Dr. G.
Yeah. All right. So I'm going to address this because I want everybody to be satisfied. This is actually very important for me, and I've been here so many years in this school, So I want really to personalize, individualize the needs of each of you. At 12:30, I'm going to invite the coordinator. So the coordinator is coming to the class, and they are going to just hear all your concerns.
Thank you.
Okay?
Yeah.
So I
Thank you,
want
Dr.
to-
G.
Thank
Yeah,
you, because I think it's important for us because we don't want to move forward like this and having to scramble and figure things out. And then it becomes a problem with our, with time frame as to studies and stuff like that, because it involves everything. But
Okay.
yeah, I feel that it needs to be heard.
All right. So in my case, for example, Ms. Pugh,
Yeah.
my mistake was not to tell everybody about the sequential and free, right? So that was what other mistake I have. What other things I have wrong?
Oh,
I
no,
want
we're not saying it's not your mistake.
No. I want to hear
general.
because I want to be everybody be open.
In
Yeah.
my opinion, I
So
think
it's-
your teaching
not-
style is very good. Like I understand it very well. I think it was just the sequential thing for me.
Okay, so that's what I was trying to compensate that. If I make a mistake, I always know I need to pay.
Right.
Thank
Okay,
you.
so and I'm very strict with myself too, and with you too, but I'm very flexible too when it's needed. So again, I'm not a monster. Again, everybody, okay, I'm not a monster. I'm here to help you. I'm here to just guide you. And some of you already, mostly all of you, mostly are being in my bioscience, so you already know how is my way to deal with these difficulties. Okay, all right. So let's do that, and let's not wait more time. Let's keep going with our lecture. Okay.
Dr. G, before we move on,
Yes, Michelle.
I just want to make sure that, because I was a little bit confused with the homework right now. I wasn't sure if it was due last night or because I went on Moodle and it said it was due today before midnight. So that I just want to clarify that it's always going to be due before the quiz, right? And
Before
not,
the quiz, yes.
okay. Thank
Yeah,
you.
I put it at midnight because I know that some of you are going to be late. So that's why I put it like that. So, but the direction is that, and I knew that this going to happen, that I knew it. So that's why I put it this extension. Okay.
So thank you for the break, Dr. G.
Sorry, there.
Thank you for giving us a break.
A break? What mean break? A break?
Like you knew that we were going to be late with our homework, so you gave us a break.
Yeah. Miss Shirley. Yes, Miss Shirley. So I'm very fair. So I'm very fair. I'm not a monster, I guess. I'm telling you again, I'm not a monster. Okay? All right. So anybody else before we start our lecture?
Oh yeah. So Dr. G, so let's say we have every week we're going to have 2 quizzes, right?
Yes. Yes,
So basically every day we have a quiz for all of our classes.
that's correct.
Okay.
Okay. And the midterm, we are going to have the Super Sunday. All right. So let's keep going. Any other question, please?
No.
Okay. All right. So let's start. Here we have this. What you see here is the ribosome. Where is this ribosome? These dots. Each of these dots is going to be this ribosome. We have 2 ribosomes, one here and another here. So here is the RNA messenger. And this is the other RNA. So this blue, I will say this blue, this is a ribosome. Another blue, another here is a ribosome. Another ribosome here. So we have different type of ribosomes. This ribosome is the cart of the Safeway. They actually you pulling in order to bring all your basket. So this is the ribosome I was mentioning. So these ribosomes are going to bring the amino acids that are going to be the sequence, follow the sequence of the RNA messenger. Where is going to happen this? In the endoplasmatic reticulum. Endoplasmatic reticulum. You okay with that?
Yeah.
Yes.
Okay. So here we have our friend, the mitochondria. These mitochondria is in the cytoplasm, is part of the organelles, and here is inside of these mitochondria where we are going to form the ATPs. This mitochondria is basically is going to be the factory to produce ATPs. The ATP is the like rechargeable battery. The ATPs is the adenosine tri, means 3 phosphate. They're going to have adenosine and 3 phosphate. 1, 2, 3 phosphates. This phosphate is the energy that we use for all activity that we have. You're thinking, you're writing, you are saying something, thinking, that you're using ATPs. And these ATP are used when you're going to get rid of one phosphate, and that produces a lot of energy. So now is ADP, because they have 2 phosphates only. So this is how we are using our energy, based on ATPs. The food and diet, the energy from the bones, that energy is not going to be used directly for us. This diet, bones, when you break down the molecules of the food, that energy is going to be used to bring a phosphate to put it back into the ADP. So the energy of the food is to put phosphates into the ADP. And then after that, and just after that, we use the ATP as energy. Is that clear or no?
Yes.
Everybody got it? Please.
Yes.
Okay. So second, here we have inside, it's going to happen basically the Krebs cycle. The Krebs cycle is the way that the body is going to produce energy. Okay, perfect. So these mitochondria are going to be in every single cell of your body. Obviously, the number of mitochondria are going to be variable according to the organ. For example, one of the highest number of ATPs in the cells are going to be located in the cells of the liver. Liver is the huge, big factory of the body. Factory because they have, how many items are going to produce the liver? 512. Imagine a factory producing 512 products. That is the liver. So if the factory is going to produce so much production of things, obviously they need more energy, right? A factory who produce more, they need more energy. And that's why the cells of the liver contain very high number of mitochondria in the cells. But the one who contain the highest, more than the liver, guess who? Is the ovum. The ovum is the one who has highest content of mitochondria. You okay with that? Okay. So, in addition to that, in order to happen Krebs cycle, you need to have oxygen. That's why we need oxygen. The oxygen is the trigger for the reactions to produce the ATPs. Without oxygen, these reactions will not happen. Okay? We have the lysosomes. The lysosomes are small organelles that are located in the cytoplasm of the cell. And these lysosomes, they have enzymes. When you have a cell here, for example, let's make it the neutrophil. Neutrophil is a round cell. This is the neutrophil. The neutrophil are going to have granules. Those are actually enzymes who are lysosomes. When the neutrophil is going to eat a bacteria, the bacteria is getting into the cytoplasm of the cell, and the lysosomes are going to come and destroy the cell, the bacteria, are going to destroy the bacteria. So those are what we call proteolytic enzymes, Proteolytic, that means that they are going to destroy the proteins of the cell membrane of the bacteria, producing the destruction of the bacteria itself. You okay with that?
Yes.
So, and please, I don't want you to think that the bacteria is getting inside the lysosome. No, when the bacteria is getting inside, this lysosome is going to open. It's going to open. And they're going to release the enzymes to kill the bacteria. Okay? Okay, so here we have the centrioles. The centrioles are basically it's going to be associated with the cell division. Cell division. So here we have the normal cell here, normal cell. They're going to then, the centrioles are somehow here, centrioles. And what happened is when the cell are going to start dividing here, they're going to open the nucleus, they're going to have the chromosomes. Then the centrioles are going to be activated. The centrioles are going to be here, they're going to be here, one, and they're going to multiply, and they're going to be another centriole here, on the extremes, on the poles of the cell. These centrioles then, can you see these centrioles like a bar, like a bars? Can you see like a lines? These centrioles are going to lace each of the chromosomes with these bars. And then they are going to pull back in order to make the cell division. So they're going to pull the chromosomes to one side, to another side. At the end, you have two cells with the same number of chromosomes. You okay with that? So conclusion here, the centrioles are going to be activated in the cell division, where especially in the anaphase stage, anaphase, we have the anaphase, they're going to separate the chromosomes to one side, to another side. Who is going to pull these chromosomes to one side, to another side? The centrioles. The centrioles are going to participate in the cell division. Parentheses, anaphase. You okay with that?
Yes.
Yeah.
Excellent. All right, so which organelle is known as a powerhouse? Who can tell me?
Mitochondria.
Excellent. Mitochondrium is not misspelled, so they're being writing in both ways. That's fine. That is the same. All right, surface organelle. The surface organelles are going to be the cilia and the flagellum. Cilia, where we have cilia. Cilia, we have, for example, this is the cell. I'm going to elaborate the cells here. Cells here, cell. And the cells are, on the top, they have like a hair. Imagine you are a cell, and your hair is the cilia. Okay? This cilia is going to basically increase the absorption or secretion. So what does it mean? For example, this cilia you see here, you will have in the respiratory tract. Remember the pseudostratified epithelium? Remember the mucus and the cilia? Remember that or no? Hello?
Yeah.
All right. And the other where this is located is in the GI tract. In the GI tract, you will see that this cilia is going to increase the surface of absorption of the nutrients, the monomers, the monosaccharides, the phospholipids, the fatty acids, and the amino acids. So that is the cilia. The flagellum is located basically in our friend, the spermatozoid. The spermatozoid is the flagellum is the continuation of the cytoplasm. The cilia is a continuation of the cytoplasm. So I will try to make, if this is the cell membrane of the cell, this is going to be, I'm going to try to draw it like this. Cilia, small projections of the cell. On the top is like the hair of the cell. And each of these are going to increase the surface of absorption for the nutrients, like mono, fatty acids, and amino acids. You okay with that?
Yes.
Okay. Yes. Now, here we have the cellular diversity. We have different size, different shape, different function. The size, the largest cell will be, for example, is going to be the ovum, that is 0.1 millimeters. So you don't need to know the number, but just remember the largest cell in the body. We have cells like, for example, the red blood cells. The red blood cells are going to be 70% of the size of the ovum. So you cannot see the red blood cells anymore because it's smaller than that. So the size is going to change. The shape. So please, I want you to remember this. We have here a cell, normal cell, typical cell. But what about if I squeeze here the cell, I stretch out the cell here, stretch out the cell here. So at the end, I have a cell like this. Which cell is that? It's a nerve cell or no?
Yes.
So this is basically a cell. This nerve cell is going to contain the same cytoplasmatic membrane, the same amounts of cytoplasm. These elongations are going to be the continuity. It's like you have your house and you have a long corridor. So the corridor inside your house is the cytoplasm. So that is a nerve cell. So we have different size and different shape of cells. We okay with that? Okay? All right.
Yes.
Excellent. So thank you for participation. The function of the cell depends, of course, right? The liver, the spleen, the lungs, the cells of the nervous system, the muscle, the bones, etc. So they are going to have different functions according to the location. Okay, protein synthesis, cellular substances that direct the production of proteins. So, for example, chromosome, protein synthesis. So please, I want you to be aware of this. Some people get confused with the word synthesis. Synthesis means you translate in your mind because there is many questions in the exam that are going to use the word synthesis. Synthesis means production. You just put it in your mind. You put it in your mind. What you're going to do, how you read that synthesis, you translate as production. Produce is going to make something. You okay with that? So the protein synthesis means the production of proteins, okay, are going to be based on the DNA. The DNA, we have the double helix, and here inside we have the recipe for the proteins. So we have genes. A group of pieces of the DNA is going to be called a gene. And each gene is going to contain a recipe for a protein. A recipe for a protein. Okay? So basically, there's a cellular substance that directs the production of protein. So for example, we have the genes. The genes. Chromosomes are going to be, what is a chromosome? If you cut in pieces the DNA, Noah and genes, pieces, big pieces of the DNA, you will have 46 pieces. You just remember, if you, for some magical procedure, you take one DNA from the cell and you stretch it out, you have 6 feet long DNA. Can you imagine that? Amazing, right? So if you cut this line in pieces, big pieces, 46 pieces, each piece represents a chromosome. And each of the chromosome, inside the chromosome, you have multiple genes that are going to be participating directly in the production of proteins. One gene equal to, say, production of protein. One gene, whatever gene you are talking about, that is the one who produces a synthesis of a protein. That protein, we already learned in the past, it could be an hormone. Protein is an hormone. Protein are enzymes. The proteins that are in the cell membrane, those are coming from the recipe of a gene. You okay with that? All right? Okay?
Yeah.
Yes.
Okay, perfect. All right, so nucleic acids. You need to be familiar with the nucleic acids, and I don't want you to get confused with nucleic acid. Nucleic acids is the big name, is the big umbrella of name for DNA and RNA. Okay, so what is DNA? A nucleic acid. What is RNA? A nucleic acid. What are the nucleic acids? DNA and or the RNA. Okay, so and these DNA, RNA are going to, remember the DNA have double helix, the RNA is only one helix. So the DNA is going to contain the A, G, and CT. AGCT. So ATCG. So adenine, guanine, cytosine, and thymine. That is the DNA. But the RNA do not have thymine. They have the uracil. And just remember the mnemonic when they're going to match AT and T and C and G. So if you have in one side of the DNA A, in the other side you have T. If you have C here, you're going to have G here. And opposite, if you have G, you have C, whatever. But always G and C are going to be together. A and T are going to be together. Remember the mnemonic AT and T. Easy. If you have A, you have T. If you have T, you have A. If you have G and C, C is the cytosine, and G is very similar to, say, cytosine with this only difference. So C and G are going to be always together. You okay with that?
Yes.
Yes.
Okay. All right, so the DNA. The DNA is, we are going to have the properties of the DNA. They are able to replicate itself. So it's a complete DNA. Why I say that? Because I want you to differentiate from the viruses. The viruses can have DNA and RNA, but those are an incomplete DNA. So that's why the virus, if you isolate the virus by itself, the virus will not be able to reproduce because it's an incomplete DNA. They cannot replicate by itself. So that's why a virus is not considered a living organism. Is that okay?
Yeah.
But you said, some of you are still telling me, but so the virus can replicate. Yes, they can reproduce, but only when they invade a host cell. The invasion is because they want to take that piece that is missing in the DNA of the virus. Okay? And the definition of to be alive is that that cell need to reproduce by itself without any help of anybody. You okay with that? That is considered a living organism. You okay with that?
Yes.
Yes.
Okay, so the DNA is able to replicate itself. When is going to replicate? When you have a cell here and they're going to go into mitosis, this is going to give two cells. And for have two cells identical to the previous one, this DNA, they need to duplicate in order to divide it in two later on, to have the same DNA at the beginning. Okay? Is that okay?
Yeah.
Yes.
Yeah.
Thank you. Then we have the mutation. Mutation, you know, mutation is very simple. Mutation you have, for example, you have A, T, C, G, T, G, whatever. So mutation means that one of these bases, these are bases, remember, are going to change position. For example, instead to have A, T, C, G, T, T, G, are going to have A, T, A, T, G, C. Mutation. They are going to change. So it's like you are changing the recipe of the ingredients of the food you're going to cook. What is the resultant? If you put salt instead of sugar, it's going to be totally different taste, or no? Totally different protein. And most likely you will not eat it. So the protein in that case will not be functional because it's not actually the shape that should be. You okay with that?
Yes.
Yes.
And it's going to store information, storage of information. The storage is the genetic passing the characteristics from mother to kids, right? To offsprings. So they're going to store actually information that is going to pass from one generation to another. So the properties of the DNA, please open eyes, open ears, is going to the replication, the synthesis, replication, synthesis, the mutation, and store information. Okay? So that is the four functions that you need to remember for the DNA. What three types of RNA are active in protein synthesis? Okay, so we have different type of RNA, by the way. The messenger we have the RNA that is the transfer RNA, and the ribosome. So remember the cards that or ribosomes that are coming with the ingredients, the cards. That is the transfer RNA. The ribosomes that are located in the endoplasmatic reticulum, that is the ribosomal RNA, and the messenger RNA that is coming from inside the cell, the nucleus. So from this, I don't want you to get confused on that. The only thing I'm going to ask and the only thing I'm going to request you to know is the role of the RNA, transcription and translation, period. Okay?
Yes.
Yes.
Okay, perfect. So we have ATs, make two, okay. The ATs, all right, so ATs, so when you have, for example, one here, the DNA, and you have A, you have T here, you have C, you have G here, you have, I will say, T, and you have A here in front of each other. It's like a zipper, the zipper of your pants, the zipper of your jacket. So this zipper, what is going to come together here is going to be molecules of hydrogen, 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 hydrogen. So that is what is going to make link one side to another side. So they are going to be linked by bonds of hydrogen, hydrogen. You okay with that?
Yes.
All right, so if I have the primary here, this is RNA or DNA?
DNA.
DNA, right? Because we don't have U, right? The U is present in RNA. T is absent, the timing is absent in the RNA. In the DNA, we have the T without U. Okay, all right. So can you see what could be the complement of A?
T.
C, G, G, C, C, G, G, C, T, A, T, T, A, T, A, C, G, etc., etc. What is the bond between bases? This is a base, and this is a base. What is the link between them?
Hydrogen bond.
Is the hydrogen. Hydrogen. Okay, perfect. All right, so you can tell which if the nucleotide on the DNA strand read CGAT, what it will be the corresponding RNA strand?
B.
It's going to be G,
C.
C,
T. U.
U. Excellent. You got it. Because the RNA do not have T, they have U instead. Okay? It's not, here is no answer.
No, Dr. J.
Okay, so let's put D here. G, C, T, U. All right, so talking about cell division, the cell division, we have meiosis and mitosis. So where is happening mitosis? So mitosis are going to be in the somatic cells. Somatic cells. Meiosis are going to be the sex cells. Mitosis, when you cut your finger with a knife or with injury, a stone, or whatever, I don't know, whatever, you injure your hand, you have a wound, an open skin. So that cells who are being destroyed, they need to be replaced. That replacement means that the cells who are still around, healthy cells, are going to start to divide in order to regenerate, to repair, to heal, to replace with new tissue, yes or no? And that is mitosis or mitosis. Mitosis is the cell division. So where is happening mitosis? Mitosis happens all over your body, except in the reproductive organs, except in the ovaries, except in the testicle. Because in the ovary, they are going to elaborate the ovules, in the testicles, the spermatozoids, and the ovules and spermatozoids are going to be divided by meiosis. sex cells. You okay with that?
Yes.
Yes.
Excellent. Stages of mitosis, we have the prophase, the metaphase, the anaphase, and the telophase. That's all what I want you to remember. But there's probably one thing. When you have problems of some genetic disorders, especially let's talk about Down syndrome. Down syndrome. Normally the cells are going to have they're going to have 46 chromosomes here. They're going to multiply at the time of the cell division during the fertilization, after the gametes, after the spermatozoid and the ovum come together, they're going to have cell division. So there is a problem in Down syndrome. So when the cells start to divide, they need to be pulled back to produce two cells. Two different cells, with the same name number of chromosomes. But in Down syndrome, that is not going to happen. When they pull the cells one to another side, this actually, one of the, when they're pulling, they're going to have an extra chromosome here and a missing chromosome in the other side. So instead to pull equally number of chromosomes side by side, they're going to pull an extra chromosome from the other side. You okay with that?
Yes.
All right. So, and that is going to happen on the anaphase. Anaphase. The prophase. What happened? This is in the prophase. Just to make a summary here, what happened in the prophase? In the prophase, this is the normal cell with the DNA. It said, "Oh, I need to multiply." Okay. So the prophase. What is going to happen is this. They are going to disappear the nucleus membrane, and the DNA start to cut in pieces into chromosomes. Then in the metaphase, the chromosomes are going to be aligned in the equatorial line. The centrioles are going to come taking his work, and they're going to start to pull to the poles, to the extremes, one side to another side. So the anaphase, you're going to have this cell like this. And the telophase is the totally cell division already. Okay?
Yes.
Yes.
Excellent. Thank you for your participation. And one more thing is that the chromosome, how many chromosomes we have? 46 chromosomes. All your cells of your body are going to have 46 chromosomes. The spermatozoid and the ovum, they contain only 23 chromosomes. But the rest of the body, 46. That are called diploid, diploid, and the other is haploid, half. Okay?
Yes.
Yes.
Okay, so now, thank you. We are going to talk about the membrane transportation. Shirley, can you tell me, or somebody tell me 12:30, please, because I don't have a watch in front of me.
Yeah, it's 12:01.
Okay, so we have half an hour. We okay?
Want to take lunch?
No, at 12:30 we have you can take, well, we need to do a give and take a little bit here. So we was talking about some difficulties, so Mr. Verdad, coordinator, is going to be here at 12:30. So I planning to do, because I don't, how many slides I have? Because you're going to take time for discussion. It's going to be at least half an hour. And I don't want to rush. We okay with that?
Yes.
We okay with that or no? So what you want your lunch break and rush at the end?
No.
So you can take your lunch in the time too. So please. So I cannot make unless you want to stay with me until 3 o'clock, I will be happy.
Can we take lunch now, like 20 minutes or something? So we won't rush in the end?
Okay, so all right.
We have tutor as well for those.
Okay, I will give you until 12:20. Okay? You okay?
Yeah.
Okay.
Thank you.
Okay. Hello, hello.
Hi.
Okay. All right, so let's wait until Mr. Please, if Mr. Verda is coming anytime because I don't see the screen, let me know, please, okay?
Okay.
Yes.
Excellent. All right, so we was talking already about the membrane transportation, right? And we have passive transportation, active transportation. Passive is do not need energy. Active, we need energy. We have simple diffusion. Simple diffusion. I'm going to make a summary here. We have the facilitate diffusion. And we have osmosis. Simple diffusion is going to go towards the concentration gradient. Towards concentration gradient. So towards the concentration gradient means that they have passing the nutrients or elements are going to pass from high concentration to low concentration. So that means you have a lot of concentration here outside, and there are going to be few concentration here. So that means that the passage of the nutrients, or the best example of simple diffusion will be oxygen and oxygen and carbon dioxide that can go out. So carbon dioxide is accumulated inside the cell, is lower concentration outside, carbon dioxide go in the opposite direction. But just make an example of the oxygen. The oxygen is low concentration inside the cell and high concentration outside of the cell. So that's why it's going to go from high concentration to low concentration. That is called towards the concentration gradient. Same thing happened with the facilitate diffusion. The oxygen is too little that they are going to pass between the cell, between the phospholipid without any problem because they're very small. It's a molecule of oxygen. But the facilitate diffusion, they need, this is the cell membrane, and here we have one protein and another protein. This is outside, this is inside. And here we have high concentration, and here we have low concentration. So that is going to pass through this protein here through facilitate diffusion. So high concentration of nutrients outside, low concentration of nutrients inside. So they're going towards the concentration gradient, but using a protein that is called, in this case, the facilitate diffusion. You okay with that?
Yeah.
Yeah.
Okay. And osmosis is the passage of water. Sorry, I'm going to put it here. Is the passage in or out of water. Okay. So if here we have, this is the semi-permeable membrane. This is the semi-permeable membrane. Semi-permeable membrane. This semi-permeable membrane, semi-permeable means they are going to permit, to allow only certain size of molecules to pass through the membrane. If the molecules are too big here, they cannot pass. But you know the concentration in the universe should be equal. The concentration inside the cell and concentration outside the cell should be equal. So normally what you want to do is that this will pass through the cell to make equal concentration in and out, but they cannot pass. So what is going to happen? Water started to dilute. So water go to the area of high concentration to dilute that area. So that is osmosis. We okay with that?
Yes.
Again.
Mhm.
All right.
Yes.
So if we have, for example, a cell here, and here we have hypotonic. This is inside, this is outside. Hypotonic, and the cells are like this, hypotonic. So we have high concentration inside the cell. Hypotonic, hypo means low, tonic, concentration. When you say hypotonic, isotonic is everything talking about the outside of the cell. So outside of the cell is hypotonic. So we have low number of protein or elements outside and high number here. So what happened? So if you have hypotonic, that means that this inside and outside should be equal concentration, but it's low actually at the beginning. So what they are trying to do is the molecules should be passing out, but they cannot pass because the membrane is a semi-permeable membrane. Do not allow big molecules passing through. So what is going to do? Water start to come in. Water start to come in to dilute it. To dilute it. If you have your tea with sugar too sweet, you put more water, right? So you try to dilute it what is more concentrated to make it equal at the end. You okay with that?
Yes.
Yes.
In hypertonic, you have high concentration outside. Hypertonic is when talking about outside the cell. And here we have some concentration. So it's called hypertonic because the concentration outside are higher than inside. But these molecules, because it's a semi-permeable membrane, cannot pass. Water starts to dilute it, and the water go to the higher concentration, go outside. Okay? So conclusion here very simple. Whatever is going to be high concentration is going to receive water to decrease the concentration. You okay with that?
Yes.
Yes, Doc.
Excellent. And isotonic means the concentration inside are going to be the same concentration outside. So there is no need to pass water. There is no need to change the concentration because the concentrations are equal, and that is called isotonic. Example of isotonic, we have the dextrose 5%. Dextrose 5%, that means glucose. Dextrose is equal to, say, glucose. Huh? This fluid that you have IB in the patients, that is isotonic. So that means that when you apply that into the patient, there is no shift or change of water from inside or outside, from inside to outside or from outside to inside the cell. And that is what because if we do this, they can produce problem. If you give hypotonic, that is water, plain water. Water is not dextrose. Huh? So dextrose, 5% means is water with 5% of glucose inside. That is the concentration of that fluid that you receive. Water do not have solutes. Water is just plain. It's totally hypotonic. It's very low concentration compared to the cells. So what happens if you give water to the patient? You can kill the patient, and the water start to get inside the cell, getting swollen, and the cells are going to be destroyed. And that happens, for example, in the nerve cells. The nerve cells, they're going to blow up, and you die from coma and cerebral, destruction of the cells. If you have hypertonic, if you give too much concentration, water start to escape from the cell, and they are going to shrink. That is called crenation. When the water starts to go out from the cell in this hypertonic solution, all this is going to shrink because it's less water, and that is called the crenation. Okay, we got it?
Yes.
Okay. The active transportation, we have the endocytosis and the exocytosis. Endo means inside, so everything that is coming in to the cell. Exocytosis is everything that is exo means exterior, outside of the cell. Endocytosis is going to divide in phagocytosis. Phago means eating. Cytosis, process of the cell. So the cell is eating. Pinocytosis, is when they incorporate fluid inside the cell. So everything is getting inside, fluid and solid. Phagocytosis, solid. Pinocytosis, fluid. So are actually eating and drinking. That is so are you phagocyte? You're not phagocyte because you're not one cell. You're eating. You have pinocytosis, you're drinking water. That is, you're not a cell. So this pinocytosis is drinking, the equivalence in us with when you are drinking. Exocytosis is everything that is eliminated, like pee and poo of the cells, for example. That is the exocytosis. Okay? So as explained before, we have the isotonic cells are not affected. So that means that there is no water in, no water out. No water in, no water out. No water in, no water out. You okay with that?
Yes.
Hypotonic is going to be, remember, you have here the cell. This is hypotonic. You have 1, 2, 3, 4, 5, 6. And here we have 1, 2. So hypotonic means low concentration outside. So what is going to happen? The water is going to go into the highest concentration. So water is going to go to the higher concentration area. And that is going to produce what I call the hypopotamus. Hypopotamus. Hypopotamus is big, swollen. Hypotonic produce hypopotamus cells. So they are going to get swollen, and it's a way to remember. If you have hypertonic, again, hypertonic is high concentration outside of the cell, but low concentration inside the cell. So, and I told you, just remember, where it's more concentrated, that is where is coming water. So water is coming from where? From the cell. So here's the opposite, see? And that is going to produce what we call the crenation. There you are, crenation. Okay, so in hypertonic solution, that is going to do crenation. We okay with that?
Yes.
Yeah.
Just to parenthesis here, apoptosis. What is apoptosis? Apoptosis means the sorry?
Cell death.
Cell death. Excellent. It's a program of cell death. So everybody have a program in your body. You know, the body knows when you're going to die. So it's time to die. That is so the cells is called apoptosis. Okay? Just to give you an example here, we have 5% dextrose, that is isotonic. We have 0.9% sodium chloride, that is isotonic too. Isotonic. So please, you need to remember this. 5% of glucose or dextrose, this is the same, is telling you that you have in 5 grams in 100 mLs of water. That is 5%. 5%. 5 per 100%. 100%. So you need, that is telling you very more important information that you have 5 grams. I didn't say milligrams, kilograms, or kilos. I said grams. 5 grams of glucose in 100% in 100 mLs. We okay with that?
Yes.
Excellent. And here we have 0.9%. That means that you have 0.9 grams into 100 mLs. Okay?
Yes.
Yeah.
So please, this is very important when you're going to do calculations for metrology. Here we have the alveolus. You know what is alveolus already. Here we have the oxygen, high concentration of oxygen, but here we have when the venous return, remember the IVC, the right atrium, the right ventricle, the pulmonary artery, then they go to the lungs, and these are branches of the pulmonary artery here. The pulmonary artery is going to carry deoxygenated blood, as you remember. So low concentration of oxygen, low concentration. Here, when you breathe in, you have high concentration of oxygen. Breathe in, air, high concentration of oxygen. But the blood that is coming back from the body after deliver the oxygen, you have low concentration of oxygen. So what happened? So they found this high concentration, low concentration, and that's how the oxygen is going to pass through simple diffusion. So again, so summary and review from the previous slide. The best example of simple diffusion is gas exchange. What is gas exchange? Oxygen, carbon dioxide. The best example for facilitate diffusion are the absorption of nutrients, the monomers, the amino acids, the fatty acids, and the what else? And the monosaccharides, the glucose, the fructose, and the galactose. You okay with that?
Yes.
Yes.
Okay. So
Yeah.
here we have cell aging. First of all, just remember the free radical. Oh, free radicals is very bad. Free radicals, what is the opposite of free radicals? Free radicals are the antioxidants. Very important, right? Remember the antioxidant. Take your antioxidant. What is the antioxidant? Orange juice. Orange juice, fruits, vegetables. They have acai. Acai, that is one of the highest components of free antioxidants. The best antioxidants are the fruits and vegetables who are very bright colors and dark colors. Remember that? And the free radicals are going to be some extra electron that is going to burn the cells. It's going to destroy the cell. And that free radical are going to be swept out by the antioxidant. So more free radicals, more alcohol, more sun, more spices, more stress, more everything produce a lot of free radicals. You get older faster. All right, apoptosis is the other name I want you to remember, is the cell programming death cell program, right? So they know when the cell is going to die. Okay, so this is a liver, normal liver, and this is cirrhosis. So cirrhosis. So what happened here in cell cancers? You need to remember that the main cause of cancers are going to be a chronic inflammation. Chronic inflammation, you remember the 5 cardinal signs of inflammation. 5 cardinal signs of inflammation will be heat. We talk about that, right? Yes or no?
Yes.
Yes.
Heat, redness, edema, that means swollen. What else? We have pain, excellent,
Loss of
and
function.
loss of function, right? So listen, if you take people who are alcoholic, they can have liver cancer. Alcoholic for 10, 20, 50 years. Inflammation means already cell damage. Inflammation means that when you say somebody is inflamed, or some cells are already dying. So these cells, they need to be replaced for new cells. If you take alcohol one time, you get drunk, okay, you're happy, whatever. The next day you don't drink anymore, so your liver gets inflamed. So some cells are being destroyed, yes. But then the liver is going to replace these normal cells, these cells who are being destroyed with normal cells, and then stop. There is no more repair because it's already healed, your liver. But if you still drinking alcohol every day, every week, every year, for 10 years, for 50, so your inflammation never stopped. So you need to regenerate again all the time. The cells will never stop to replace the cells that are being damaged. In that process, the cells, in some moment, they produce a mutation, because all this replication, regeneration, are being commanded by the cells. But the cells said, why, I don't need to stop because never stop. I need to replace all the time. So in the DNA happen a mutation. And that mutation means that the cell they are going to forget how to stop multiply. So they're going to multiply without a stop. Even if you stop already the drinking, it's too late probably at that time, you're going to still reproducing cells. Cells reproducing without a stop produce masses, tumors, cancers. You okay with that?
Yes.
Yes.
Cancer risk factors. We have hereditary, so there is some genetic component. So there is cancers. Cancer basically they need, you have a risk to have cancer, okay, but that is going to be hand by hand with the environmental surrounding. So environmental factors plus genetic factors are going to play a role together. You can have cancer, you have high risk for cancer, but you are not exposed to environmental problems or injury, you will never have cancer. Example, if you have fair skin, you are very white, you have risk to have skin cancer. Two siblings are going to have very white skin. One develop cancer, the other do not develop skin cancer. Why? Probably because one that developed skin cancer was exposed to the sunlight more than the other one. The exposure to the sunlight is what we call the environmental factors. We okay with that?
Yes.
Okay.
Dr. G, is Mr. Verdas still getting on the call?
Is here already?
No, that's what I'm asking because
Yeah, please. Yeah, thank you. Let me know. I need to finish this. So I send invitations. She should be here anytime. Okay. All right. So please, I want you to consider, please, that I need to finish this class too. Okay. Okay. So we have 4 types of tissue. We have epithelial tissue, connective tissue, muscle, and nervous tissue. Okay? So how many types of tissue we have? 4. For this, I want you to remember epithelial. Epithelial is going to be basically all this tissue. Simple squamous, etc., etc. I'm not going to go that. The one I want you to remember is this one, the only one here. The pseudostratified epithelium. And that is review from bioscience. So let's go to the pseudostratified epithelium. The pseudostratified epithelium, this is the respiratory tract. Here we have this pseudostratified epithelium, They have a cilia here. And actually, they have here, some cells are going to differentiate and transform into goblet cells. These goblet cells are going to produce mucus. Okay? So that is because when you are smoking or you have some smoke, whatever, injury, the cilia, what they are going to destroy is the cilia. And the cilia is moving, trying to get rid of the bacteria that eventually can get into your respiratory tract. So when you go into the respiratory tract, you have the pseudostratified epithelium with the cilia and the mucus produced by the goblet cells. And the bacteria, when it's getting into your respiratory tract, is getting like in a swamp. So they cannot keep traveling deeper into the respiratory tract. So, and the cilia is moving like a broom, taking out of all the bacteria and all the dust that is coming, and the bacteria and the mucus. The mucus is going to delay the traveling of the bacteria, giving time to the cilia to super out. You okay with that?
Yes.
Yes.
All right, so epithelial tissue is going to be basically all the lining, outside of the organ and inside of the organ. Epithelial tissue will be, for example, your skin, your nails, your hair, your eyebrows, so all hair, and all the internal lining. Put your finger inside your mouth, put your finger in your esophagus, put your finger, what you touch, the lining of the stomach, or the gallbladder, or the urinary bladder, all the arteries and veins, the lining is epithelium, that is called endothelium. Okay, I'm not talking something new. Huh? This is something that we already know. So this endothelium is an epithelial tissue. So everything that is hollow organ, the outer and the inner lining is epithelium. You okay with that?
Yes.
Yes.
Epithelium is going to include the glands. Glands, we have the sweat glands. We have the sebaceous glands. So we have different glands in your body, including the thyroid gland. the adrenal gland, pituitary gland. All these glands are epithelium. Okay, so epithelium tissue corresponds all the lining from hollow organs in and out, covering, and the glands. Glands include the exo and the endocrine glands. We okay with that?
Yes.
Yes.
By talking about the connective tissue, very simple. I want you to remember here, we have the loose, and well, I want you to remember liquid, structural. So liquid is the blood, connecting all the 11 systems. Heart, bones, and cartilage. Then you have the soft, that is the fat, loose fat, that is basically under the skin, whatever, right? Covering organs, so loose fat. And the other one is the fibrous. That is what you need to remember, fibrous. Ligament and tendons. Ligament and tendons are going to be fibrous connective tissue. Open eyes, open ears, question for the exam. And just remember, what is the function of what is the function of, let's go back. Connection, what is the function of the epithelial tissue? To cover. To cover. What is the function of the connective tissue? To connect. Simple. You okay with that?
Yes.
Yes.
Talking about the muscle, we have the smooth muscle, the skeletal muscle, and we have the cardiac muscle. Talking about the skeletal muscle, skeletal doesn't mean, it's not the bone. It's the muscle attached to the bone. The skeletal muscle is called the the skeletal muscle is the voluntary muscle, or striate muscle. The smooth muscle is going to be the involuntary muscle. And the cardiac muscle is the cardiac muscle. It's just like that. It's actually involuntary too. Can you stop your heart? No, right? It's involuntary. So you cannot do that because it's involuntary. It's going to work automatically. A skeletal muscle, just remember, we have basically, this is cylindrical. This is more elongate cell. The skeletal voluntary striate muscle, they have multiple nuclei. A lot of nuclei everywhere, like a potato. It looks like a potato. And here we have the smooth muscle, is only one nucleus. But here in the skeletal muscle, we have striations here. That's why it's called striate. And what is going to contain these striations? They're going to contain the proteins. Which proteins that we talked before is the myosin and the actin. Myosin and actin. Meantime, the smooth muscle do not have striations. Do not have those striations. I left the cardiac muscle at the end because the cardiac muscle is having something similar from one to another. Let's talk about the cardiac muscle. The cardiac muscle are going to have this is a square cell. It's like a square cell. They have only one nucleus, similar as the smooth muscle. But they have some striations, similar to the skeletal muscle. See, the cardiac muscle have similarities between the smooth and the striate muscle. So that's why I put it at the end. And in addition to that, we have these strong connections. They are going to connect to another cardiac muscle here. And these strong connections here, these and these, those are going to be called the intercalate disc. Intercalated discs. Okay? Intercalated disc. So what is the function of the intercalated disc? Is to connect one cardiac cell to another cardiac cell. Why? Because they need to very efficient to pass the electrical impulse that make your heart contract and relax. So that's why they have very intimate, very close connection through these intercalated discs. These intercalated discs are not present in the other type of muscle. We okay with that?
Yes.
Yes.
Okay. Now here we have the nerve cell. You already know the nerve cell is a cell, right? So cytoplasm, etc. So here we have the nerve cell is going to be composed by the body or soma. Question for the exam. Body or soma. Body or soma is all this. This is the nucleus. The nucleus is included in the body or soma. Soma. The nucleus. So from this, can you see the cell membrane of the cell? Cell membrane. Look at this cell membrane. This is cell membrane or no?
Yes.
Tell me, this is cell membrane or no?
Yes.
Again.
Yes.
Can you tell me this lining cell membrane or no?
No.
Yes.
Yes. See, you see, the cell membrane is a continuity here. See? So the dendrites are the continuation of the cytoplasm and the cell membrane. Same here. Can you see the cell membrane here? The cell membrane is going to go all the way here. This is cell membrane. So this is like a corridor with cytoplasm inside. So this is part of the cell. You okay with that?
Yes.
Yes.
Yeah.
Excellent. So now, here we are going to see the body and soma. These are the dendrites. Those are dendrites. When you study more, you make more dendrites. Don't forget that. So the more that you study, the more dendrites you obtain. And the dendrites are going to give you different pathways to get to the answer, to memorize something, to remember something. I remember because something of this, that is one example only. Because you create a dendrite that associate one thing to another. See? That is dendrite. Then we have the axon. The axon is going to be the continuation of is the part of the cell membrane, is the continuation of cytoplasm. And they are going to end in the terminal axon. Terminal axon that is going to connect to another nerve cell. So conclusion here, components of the nerve cell is the body soma, dendrite, axon, and terminal axon. You okay?
Yes.
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 Then we have the glia. Glia or neuroglia. Glia, neuroglia are cells who are not nerve cells. They are not nerve cells. Here we have a neuron. Can you see here with the axon, whatever? This is the dendrite. So here this is the nerve cell, neuron or nerve cell. Here we have a capillary because the nerve cells, they need nutrition, they need oxygen, they need carbon dioxide, they need elements to survive, correct? So this oxygen and nutrients, they need to go to the neuron. But in between, we have one cell that is called a glia or neuroglia. There is different types. Oligodendrocytes, we have astrocytes, we have never. So you don't need to remember that. But there's a glia. This glia is going to give support and nutrition to the cell, to the nerve cell. So this is the blood here with the blood capillary, with the oxygen and nutrients. So these neuroglia or glia are going to have connections, pedicles are called. They're going to pass the oxygen, the nutrients, and then they pass to the nerve cell. So what is this glia? This glia is a filter that is going to prevent that entry, for example, of bacteria or toxins. So this is the barrier that is going to filter everything bad go away, and they're going to pass only everything good that is for the nerve cells. Remember, the glia is not a nerve. It's not part of the nerves, right? It's not a nerve cell. It's a cell who gives support. We okay with that?
Yes.
Okay.
Yes.
The last thing that we are going to talk is the Schwann cells. These Schwann cells. So look at this. This nerve cell is like this only, yeah? Like this. It's a naked axon. This is the axon. It's a naked axon. These are extra cells in purple. So what happened with these cells is this. This is the axon. This is a cell. I'm going to put the nucleus here. Can you see the nucleus here? This is a cell. This code is a Schwann cell. And what happened here is, this is the axon, this pen. And what happened is going to do this. They're going to do this. They are going to go like this, and they are going to go like this. 1, 2, 3, 4 times. Can you see the nucleus of the axon of the Schwann cell? Is this. So you have 1, 2, 3, 4, and many of them, one after another. These are going to be called the Schwann cells. Okay. This is the nucleus of the Schwann cell. This is the axon, and that is going to be wrapped around many times. So these Schwann cells are going to contain myelin. Myelin, what is myelin? Fat. It's a type of fat. This myelin contained in the cytoplasm here of these Schwann cells are going to accelerate, increase the speed of the electrical impulse. So the electrical impulse is coming here, and they are going to literally jump here. Jump, 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 jump. They are going to be very fast, very fast, very fast, very fast, very fast. But if you don't have these Schwann cells, the transport, imagine without Schwann cells, the transportation of the electric impulse will be like this. That's the speed. The difference between the speed of nerve cells contain Schwann cells and the other one do not contain Schwann cells are going to be like what the fastest. It can go from 100 yards you run in 1 second. And the cells who do not contain the Schwann cells are going to run 1 yard per second. See the difference? 1 yard per second for those nerve cells who do not have Schwann cells, but the ones who contain Schwann cells, they run 100 yards in every second. You okay with that?
Yes.
Yeah.
And we was talking about that the brain is going to contain the gray matter. Those are the sinkers. That is the cortex of the brain. Those are nerve cells without Schwann cells. But the white matter that is under, that is actually the purpose of that is to communicate, to send messages. So that's why they need to be faster. So in conclusion, the white matter have myelin cells with myelin, nerve cells with myelin, and Schwann cells, and the cortex do not, because they are sinkers. Those are the ones who give commanders. But that is what we need to remember at this moment. So axons with Schwann cells faster. Without myelin, without Schwann cells, slower. You okay with that?
Yes.
Yes.
Okay. So let's see. Mr. Verda is here or no? Yet? He's
No.
coming.
No.
All right. So I'm going to call him. Okay. Any question, please? We are going to do a review after the meeting. Okay.
Thank you.
Okay. Thank you.
Please, I want you to understand I'm trying to do my best to fix everything and to make actually.
Thank you.
Thank you, Dr. G. We
We
appreciate it.
Dr. G.
Thank you, all of you. Thank you, all of you. My concern is your concern is my concern. I'm calling now.
Dr. G, if you're talking to us, you're muted.
All right, so he's not answering yet. Probably he's finishing what he needs to do. And meantime, let's take advantage of that and let's do our tutor. You okay with that?
Yes.
You will tell me when he's coming in. For this, I need to cut my presentation.